So in section 6.2, we are going to talk about the actual process of cell division. So it's not just that the two cells just kind of decide to split in half and that's all that happens. There are actually several steps specifically involved in splitting the two copies of DNA and making sure that each new cell has identical copies of the DNA. Okay? So this whole process is called mitosis. So cell division progresses through several distinct phases. We talked about them briefly in the last section. So you have interphase, which is your general growth period. It's also when you're going to have the synthesis phase, where DNA gets replicated. Then you go straight into prophase. So prophase is the first step of actual cell division. Then you have metaphase, anaphase, telophase, and cytokinesis. So this divides what is known as a parent cell, so your original cell, into two identical daughter cells. Okay? And each daughter cell is known as a sister cell to each other. Okay? So you have the single parent divides into two daughter cells, which are sisters to each other. Okay, and this whole process is known as mitosis. So get ready for a nice little biology joke. Haha. Mitosis. Haha. Anyway, okay. So again, just brief overview of the last section. Interphase includes your G1, S, and G2 phases. Okay, so this is kind of basically what the cell looks like during interphase. Okay, and this is just highlighting the key factors in cell division. Okay, so you have your parent cell here, you have your centrioles, okay, which is an organelle that we talked about. They are useful for helping to organize the microtubules, which are going to be incredibly useful in dividing the DNA, as we'll see in a little bit. Okay, so that's why the centrioles are on here. Well, in this case, the microtubules that are used in cell division are called spindle fibers, okay, and then all together they are called the centrosome. And then we also have our nucleus with our DNA. Okay, so we have, let's see, four different pieces of DNA in this nucleus here, okay. And you can see that they are kind of just hanging out in the middle of the nucleus. There is no condensation of these DNA molecules. They're just kind of these long strands. So those long strands make kind of a mess within the nucleus. So it's just this tangled ball of basically yarn. But that is actually a really good state for the cell to go in and actually make copies of the DNA in order to use it to create proteins. So this is a perfect state for normal functions, growth of the cell, all of that. Okay, so interphase really prepares the cell to divide, replicates organelles, replicates the DNA, and then grows the cell itself. Okay, so the cell grows during both G1 and G2 phases, and then your DNA is again duplicated during the S phase. So after interphase, we go right into prophase. Okay, so in prophase, this is when you first see the DNA start to condense. Okay, so oftentimes when DNA is condensed, it is represented by a little X. Okay, we'll see why in a little bit. Those X's are known as the chromosomes. Okay, so the DNA in the chromosomes condenses. Okay, so you get this specific X structure, and it's looks a lot thicker. Okay, so you can see this is an actual image of a real cell, okay, that is in prophase. Okay, it's starting to get into prophase. So you can see that the DNA is no longer just little blue strings all throughout the nucleus. There is some thickening to these chromosomes. Okay, you can't really see the X so much because they're all on top of each other right here, but that's why we have this kind of sketch here. You're also going to have the spindle fibers forming up. Okay, so the centrioles will start to migrate towards opposite sides of the cell, and they will form the microtubules between them. Oh, wait, one more thing, sorry. And finally, you're also going to have the nuclear membrane start to dissolve. Okay, because remember, the nucleus is designed to keep the DNA separate from the rest of the cell. But in order to divide the cell, you need the DNA to be with the rest of the cell. So the nucleus, the nuclear membrane, will actually start to dissolve during prophase. Okay, so that's why it's represented by the kind of hashed marks here. Okay? All right, moving on. Now we're going to talk briefly about chromosomes. So DNA is divided into things called chromosomes. Now, the number of chromosomes in each nucleus of a cell in any organism, that number is unique to that organism. Okay, so like humans have 46 chromosomes. Okay, so each species has their own unique number of chromosomes, and they always come in pairs. Okay, so you always have multiples of 2, okay, total. So whenever you look at all of the chromosomes for a specific species in an image like this, it's called a cartograph, okay? So in this case, we are looking at a human genome. So you have your 23 pairs of chromosomes, and it goes from, they're basically just organized in decreasing size. So you have 1 being the largest all the way down to 22 as the smallest, and then you have your X and Y chromosomes as well, okay? So again, humans have 23 pairs of chromosomes, meaning that we have a total of 46. And again, chromosomes are organized based on their size. Okay, so largest first all the way down to 22, and then the 23rd pair are your sex chromosomes, okay? So X or Y. So chromosomes condense at the start of mitosis during prophase. So again, this is kind of a method of basically untangling the different pieces of DNA and then making them easier to handle. Okay, so that you don't have all of these knots forming within the DNA when you're trying to pull the strings apart. So DNA, when it condenses, actually wraps around proteins called histones. Okay, so this is basically creating a spiral. Okay, so this is different organizational levels in these chromosomes. So first up, you have your DNA double helix. Okay, this is the straight kind of string of DNA. And then when you get to your first stage of condensation, you have your histones get added in. So these proteins are basically just like, if you think about a yo-yo, okay, this is the body of the yo-yo, the histones are the body of the yo-yo, and then the DNA is the string that just gets wrapped around that yo-yo. Okay, now in this case you have several of these yo-yos kind of next to each other, and the DNA wraps around twice, or wraps twice around each one of them. Okay, and then once you have the DNA wrapped up in with the histones, then you start coiling the DNA. Okay, and that's when you get to chromatin, and you coil that as well, which is the supercoiled DNA. Okay. So it's kind of the idea of, you remember, if you know the old like landline telephones that had the wires on them that were coiled. Okay, there's actually, if you've never seen one of them, the phone in my room actually has one of these coils on it. But if you just had that cord stretched out completely flat, it would be really, really long. But because it's coiled, okay, it actually makes it look shorter. But if you have to travel, okay, with that phone, the cord will actually stretch out to a certain distance because of that coiling. So that's the same kind of concept here with the chromosomes and how they condense. And this is where you actually get the X structure of the chromosome. Okay, this is why we get that kind of image. So this is an actual image of a chromosome. Okay. And all of this, it may look solid, but that is strands of DNA wrapped around proteins and then kind of wrapped around themselves multiple times. Okay. Okay. So DNA plus proteins is called chromatin. This is where we're going to get into a lot of very similar sounding terms, okay? So just be very careful with your definitions here. So you have DNA and you have your histone proteins combine to form chromatin. Now chromatin will condense further to form a chromosome. Okay? Now, 1/2 of a duplicated chromosome is called a chromatid. Okay? Nice and annoying. So we have the chromatin that kind of gets wrapped up into this thicker structure here, and then it is a duplicated chromosome. Okay, so you have 2 copies of the same DNA here, and one of those copies is called a chromatid. Okay, so the only time you will see a chromosome in this X structure here is when it has actually been replicated. Otherwise, it just kind of looks like a wonky line. Okay. Now, sister chromatids, basically they are exact copies of the same DNA. Okay, so this is a sister chromatid to this over here. Sister chromatids are held together at the centromere. Okay, so we have the centrosome, which is part of the division process. It contains the microtubules, and now we have the centromere, okay, that holds the sister chromatids together and creates the chromosome, okay? You also have what are known as telomeres. So telomeres are, this isn't necessarily pointing to the right area, but telomeres are the end of the DNA. So they are basically just extra base pairs of DNA at the end that don't actually include any genetic information. They don't code for anything. They are just there to protect the actual DNA information inside the chromosome. Because the chromosome's weakest point are its endpoints. Okay? So basically, if something were going to attach to the DNA to try to degrade it, it's going to attach to one of the ends. Okay, so the telomeres are there to kind of, you know, give the whatever is going to attach to them something to chew on until the cell can be like, hey, you shouldn't be eating that, stop it. Okay, so a lot of times if you see like a lot of cell damage, it's happened because the telomeres were actually completely worn through. So there are no longer any telomeres and the ends are just exposed genetic information. Okay, now your sister chromatids will actually separate during mitosis, and that's how each cell gets its own copy of the DNA. Each cell gets a chromatid for each chromosome. All right, so we're going to stop here for a second and pick up with the next video.